Okay, good afternoon everyone. So yes, I was brought into this to talk a wee bit about our experiences in the biotech. I must admit I found it quite a difficult subject. It's not one that I normally talk around, but it does happen very much in the background of a lot of companies. So I will have a go. Let me introduce you to Mission first. I'll just do the build. So we're a small biotech, Cambridge-based, based at Babraham, local to here. Our core area of expertise is within the deubiquitinating class of enzymes. Sir Phil talked about the ubiquitin pathways. These are essential enzymes in those pathways. We co-founded the company with Steve Jackson from the Gurdon, of which we were all part of the Kudos team prior to coming to Mission. We've got a very good investor base, some great investors that have joined us on our journey. We've raised about £27 million to date, and we're about 4 years into the company. So we have really put together a sort of proprietary platform around the DUB area, and a lot of our chemistries and biology is around identifying drugs to the deubiquitinating enzymes, or the DUBs. Predominantly around this DDR. We're a DNA damage response company. We started Mission on the same idea as Kudos to look, as various others have talked a wee bit about, the synthetic lethal concepts in order to try and identify mechanisms where you can selectively kill cancer cells. But actually from the platform, we've really developed into a company that's spread out into a number of therapeutic areas, because the DUBs and the ubiquitin pathways, as Sir Phil said, play a role in a number of disease states. Yeah, we're doing very well and moving things, progressing things forward. So I'm here to talk to you a wee bit about, our collaborations, what we do. I'm not touching on the sort of CRO collaboration. This is more for me around the academic and the pharmaceutical collaborations that you would have within a small biotech. And these predominantly fall into these sorts of categories. Basically, for the academic side, it's often to do with, getting access to potential models, etc., or trying to get some data and studentships involved in those labs. Whereas on the pharmaceutical side, there's a lot of data transfer. Often it's around the due diligence process, and I'll talk a wee bit more about that. And that entails compound testing. And again, there are some fairly fundamental agreements, and the CDA, the MTAs are used in anger in many respects in these. But you then do have subtleties, you have research agreements, and you have student agreements on the academic side. So these are all fundamental tools for us to be able to sort of get into the data and discuss that data. However, I want to sort of give you a scenario. So you envisage where I'm standing at the bar after a conference. I've had a couple of drinks. I'm standing next to an academic. They've had a couple of drinks. And I say, oh, that was a great talk you gave this morning. And those models, it'd be fantastic if we could test our compounds in your models. Yeah, we were looking for very novel compounds. Why don't we take your compounds off you? A few more drinks, flowing now in the evening. Right, well, we'll send you those compounds. We've got very good nanomolar hits, your model's exactly where we need to be, etc., etc. So it's all trusting, it's open, it's a collaborative spirit when you have that discussion. And this is where you would like to try and keep it when you're talking scientist to scientist. However, the next day you go back and you have a chat with your legal team, and the reality kicks in. And this reality really is the thing that makes it a wee bit more difficult to achieve over your drink with your colleague, the actual realities of having that, open, collaborative style. And that really comes down to the fact that there's a certain suspicion then kicks in, and that's around, what are they wanting from those results? Who's actually going to own those results? What happens if there's IP generated? And therefore, this sort of puts what would you want to be a fast-moving collaboration in some respects, i.e., you want to get them your compound, they want to test them, and let's have a look and see what we've got, really grinds to a halt, and it can take months, and I've even seen it take years to try and get it sorted out. And it really does become very painful. So there is a reality to what you would like to try and achieve, and I think that can be quite difficult. It is necessary. So I'm going to talk about a couple of case studies. So the first one is, okay, we're a small company. We're looking for collaborations. We're looking for partnerships. Obviously, we want to carry out a due diligence, get due diligence carried out on us, I should say. And part of that will be us sending our compounds out to those large pharmaceutical companies, and let's just call it Pharma X. And there are a number of issues when that starts to happen. I mean, the first thing really is around the learning, i.e., you're sending your compound to them. What happens if they carry out, more assays, do more with your compounds than you have done? So they're going to get more of a learning on your compounds than you. And that can be quite dangerous. It means that you might have missed some IP that they've spotted, for example. Then there is actually who owns the results, i.e. they're generating results but on your compounds. Therefore, who technically owns those results, and how are you going to divvy up those results in some respects. And another one is often these initial look-sees don't lead anywhere. So you end up with, results that have been generated that you potentially couldn't use because they've come back to you. They look very interesting, but obviously they're not enough to kick off the partnership or collaboration. Therefore, what can be done with those? And that sometimes can be a bit frustrating because you're sitting on those results. And then there are other points that do kick in, and I've come across. Contamination, obviously, if it's involving structures or someone wants to see structures, then it might both contaminate them in some respects because they've seen your chemistries, therefore they can't carry out those chemistries. And it might contaminate you in some respects that they have now seen those chemistries and therefore it makes life a wee bit more difficult for you. Often this is done through a third party, in terms of a third party looking at your chemistry, and then they put a report in. And the last thing I think I've got on there is really, and I've seen this, where you've generated almost the same type of results that they are looking to do, and you haven't put your IP down in those results. So the discussion is, well, if they get exactly the same, you're now tied into this. These results are going to be, between the two of you, very similar to what you have, and it's tied into this agreement. How are you going to then distinguish your results from theirs? And it can all get quite messy. So these are the sorts of issues that can arise from that due diligence process. So in terms of resolution for some of these, and I'll just talk my way through them. For me, it's certainly about making sure that you have as much IP up front. I mean, if you can do as much as you can up front before you send your compounds, then that's important. And certainly it's around the chemical matter. If you can make sure that you've got your novel chemical matter down before you send your compound. Sometimes it comes very close to the wire, and sometimes you're sending the compounds as you're submitting the actual IP. But it is important. And then if you've actually got some biology you can capture, then that actually helps as well. Then in terms of the experiments, I mean, I've seen for due diligence is they send a list. I mean, when you give it to a scientist, and if their legal department go to their scientists and say, what do you want to carry out for due diligence? They will list everything. I want to do this, 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 this. Oh, it'd be great if we get enough for in vivo. We'll test it in that. We'll do it this year. And that can be dangerous because it just creates a lot of data that, as I said previously, you haven't seen. And examples are often in oncology is putting things on cell lines. Let's put it on our panel in-house, 100 cell lines. Well, actually, do you need to put it on 100 cell lines? Can you get away with putting it on 10 or 20 cell lines to give you a flavour of what that is actually about that compound? And it gives you enough of a warm feel that you would like to go to the next phase. So I think if you can, and it looks sensible, then see what you can do in terms of restricting the type of data generated. And then ownership of results. Obviously, what you're trying to do is for them to get enough out of the due diligence to make a decision. but you do not want them to actually then use those results or that data in any other way. And therefore, you have to have this sort of non-use provision, i.e. you cannot use these compounds for anything else. They have to only be used under the due diligence process. And that is important. So also, do as many upfront experiments as you can. If you think, for example, often what you see is you might get your list of experiments they want to do, and you think, oh, that's a good idea. Oh, we should go, here, let's go off and do that. So you do those experiments, get a value for it, and get an understanding of it, and then it means that the results you get back aren't, you know, things that potentially you haven't spotted yourself in some respects. And then, yes, so you can usually get this to restrict the results for in-house use. So basically, what will happen is the results tend to come back. You agree that these results can only be used in-house. However, if it fails, the collaboration fails, or the partnership fails, then put some sort of time clause on it, where you can actually then say, well, after X number of months, we can then use these results. And try and get that as far up the process as you can. I mean, often it might be like 12 months, but if you get it from the start of the signature of the MTA rather than from when the results are delivered, which might take several months, then at least it's that clock is ticking. And then obviously it is make sure that in the MTA you have that they cannot deconvolute or identify what your chemical matter is. So that's case number one. That's just around the actual pharmaceutical side of things. In terms of case number two, which is an academic collaboration, these are different. They have a different style. It's often when you're working with someone in a scientific way that you want to be open and, you know, be as collaborative as possible, and it's mutually beneficial for both parties, i.e. they've got an assay that you want to test your compounds in, and therefore, you know, they're going to gain results and you're going to gain an idea of how potent your compound is. So, you know, it is a bit of a win-win situation, and therefore it's important that, you know, you try and sort this out. However, there are a number of issues that do arise, and we have seen them several times. Often, again, the ownership of results comes up. You know, when I was first looking at, you know, working with academics many years ago, the tech transfers weren't as active as they are now. They are very savvy in terms of what they look for now. In terms of publications, I mean, this is the bread and butter of an academic unit, and that's important. So there will be a discussion around public, you know, putting out those data into the public domain. And then, obviously, controlling compounds in the academic environment. And I have seen this where, actually, you give a compound to one person, and we had, you know, several months later, a publication come out from someone else, and you think, hang on a minute, how did that person get that compound? And that has happened, and so you have to be wary that that could happen in the environment. So resolution, just about finished. It does take an awful long time to try and work out the details of who owns the IP, and often it is left to a rainy day, i.e. that you're not going to resolve all the issues up front, so it's often, let's put this off, let's have that discussion if there is IP generated, and often that is the case. It does make that next phase a wee bit more difficult. And then concede the publication will happen, so often you may be able to put a time delay on that, and that's the thing to try and achieve. And then I missed out the costs the companies have. They often, I think, academic groups tend to think that pharma are fairly well flush with cash, and that sometimes can be quite difficult. So I often just plead a bit of poverty. So on that note, I will leave it there.
Thank you, Niall.